In today's video, I'm going to be answering the IGCSE Edexcel paper 1B from May 2018. So starting with question 1, the diagram shows the alimentary canal of a rabbit with some parts labelled. The table lists descriptions of some parts of the alimentary canal. Complete the table by giving the correct part from the diagram to match each description. One has been done for you. So whatever you do, make sure you're picking labels from the diagram and not making them up out of your head. So used to chew food is the teeth. Yeah, that makes sense. Has most villi for absorption. Well, remember that these are found in the small intestine because that's where nutrients are absorbed into the bloodstream. So I'm going to write small intestine as my answer. Produces hydrochloric acid. That's produced by the stomach in order to help break down food and kill pathogens. So I'm going to label that stomach. Stores feces. Remember that feces is basically poo and these are stored in the rectum. Explain how the liver helps digestion in the alimentary canal. So here it's easier than it sounds. You just need to try and remember a substance which is produced by the liver, and that is bile. So bile is made by the liver. It has two jobs, remember. One of those is that it emulsifies. And that means that it breaks down large fat droplets into small fat droplets to increase their surface area for breakdown by the enzyme lipase. And remember that lipase is an enzyme. The second role is that bile also neutralizes hydrochloric acid to make sure that the pH is appropriate for the enzymes found inside the small intestine. But the easiest thing to write here is that bile also neutralizes stomach acid. C. Rabbits eat grass. The diagram shows the cell wall of a leaf cell from a grass plant. Complete the diagram by drawing and labelling other structures in this leaf cell. So we're looking for a regular plant cell and we're looking to achieve 3 marks. So handily they've drawn us the outline. Remember that there's a vacuole in the middle, a nucleus, and scattered around the edge we have chloroplasts. And now I'm going to start labelling. So the outermost edge is the cell wall. Remember that's made out of cellulose. The inner layer is the cell membrane. Here are the chloroplasts. Remember these are the site of photosynthesis. The larger structure is the nucleus. I've already mentioned the vacuole. And the jelly is cytoplasm. So we've given way more than 3 marks, but I like to be nice and detailed with my answers. D. Grass cells are made from cellulose. Rabbits cannot digest cellulose. Bacteria living in the large intestine of rabbits can digest cellulose into glucose. Rabbits eat their own feces to obtain this glucose. Give 2 differences between the structure of bacterial cells and the structure of plant cells. So you can give anything sensible here. So for me, if you remember the bacterial cell, it looks a bit like this. It has a flagella. It does have a cell wall, but its cell wall is not made out of cellulose. So you could write here, the bacterial cell wall is not made out of cellulose, unlike the plant cell, which they've already told us here in the question that it is made out of cellulose. Secondly, remember in a plant cell it has a distinct nucleus. There's no distinct nucleus in a bacterial cell. Instead, they have a circular chromosome or a nucleoid. So bacterial cell have no distinct nucleus. Instead, they have a nucleoid. Describe a test to show that a rabbit feces contains glucose. This is nice and straightforward. So we want to name the reagent, which is Benedict's. So you heat the feces with Benedict's reagent. And in terms of colour, it should turn from blue to brick red if glucose is present. 2. Human blood contains red blood cells and white blood cells. Different situations can cause changes in the numbers of these blood cells. The table shows some of these changes. So having a poor diet can mean that your red blood cell count decreases while your white blood cell count stays the same. Living at high altitude, so at the top of a mountain, for example, you find your red blood cells increase, your white blood cells stay the same. Receiving therapy for cancer finds that your red blood cells and white blood cells both decrease. Explain why poor diet can cause a change in the number of red blood cells. So how does diet link to red blood cells? Now, red blood cells, remember, contain a pigment called hemoglobin. And what is present in that hemoglobin is iron. So we know we need iron in order to manufacture the hemoglobin found in red blood cells. So poor diets, therefore, to link it to the question, contain little iron, and therefore less hemoglobin is made and fewer red blood cells are made. Suggest why athletes often live at high altitude before completing a long distance run. The clue here is that it says at high altitude that the red blood cells increase in number. So let's make that statement first of all. So why is that good? Well, that's because of the role of red blood cells. Remember, red blood cells transport oxygen. So this means that more oxygen is transported around the body. So more oxygen is transported. Why is that oxygen important? Well, that's because it's used in a type of respiration, and that type of respiration is aerobic respiration. Make sure you specify the type. And therefore, the respiration releases energy which can be used to help muscles contract, which is what you actually need when you're running. Explain how the change in number of white blood cells during therapy for cancer could be a risk to the patient. And actually, we know that from the table that the white blood cells decrease. So what is the problem with white blood cells decreasing in number? Well, think about their roles, and that's actually what this question is asking. The same question could actually be, what are the roles of the various white blood cells found within the body? So if we reduce the number of white blood cells, it means that we reduce the number of phagocytes. And what is the role of phagocytes? Well, remember that they engulf pathogens. So a decrease in phagocytes means that fewer pathogens are engulfed. On top of that means that you have fewer lymphocytes. They're the second type of white blood cell you need to know about. And remember that lymphocytes produce antibodies which attack the pathogen. So fewer lymphocytes means fewer antibodies released to attack pathogens, e.g. the influenza virus. It's nice to give a bit of context and a few examples if you can. 3a, Charles Darwin observed finches, which are a type of bird, in the Galapagos, a group of islands off the coast of South America. Darwin's observations helped develop his theory of evolution by means of natural selection. The diagram shows that the common ancestor and some of the finch species now living in the Galapagos. So you can see the large ground finch has a very large beak, the cactus finch at the bottom has a very small one, and then there's some intermediate ones in between that. The large ground finch has a large beak and feeds on big seeds with hard seed coats. That makes sense, so he can crack them open. The cactus finch feeds on nectar from cactus flowers. Explain how the beak of the cactus finch could have evolved by natural selection. So the cactus finch, let's have a look, it has smaller beaks. So this is your rote learn answer about natural selection. Remember, I write all these answers in my PDF revision guide. So for everyone that's got that, you want to learn this answer off by heart and then just trot it out here. So first of all, state that there is variation within, and you just make it applicable to the particular question being asked. So there's variation within the finch species due to mutation. This means that some finches, and let's have a look at the picture. Well, the cactus finch has a smaller beak, have smaller beaks, and can reach the nectar. So they can reach the nectar better, which means they can feed better, and therefore they get more food, they're more likely to survive. These finches are more likely to survive and subsequently reproduce. And that's the key thing when we're talking about natural selection. They must reproduce, because when they reproduce, they pass on their favourable allele to their offspring. And that really is the mechanism of natural selection. One of the Galapagos Islands is called Fernandina. It suggests why the large ground finch is found on Fernandina, but the cactus finch is not. Nice common sense question here. So what must be clear about Fernandina is to do with the food and what sorts of food it has available. So you want to say here that Fernandina lacks cacti, but has plants which produce, and let's double check, the ground finch feeds upon big seeds with hard seed coats, which produce big seeds. So it's a difference in the food supply, really. A student uses 400 clay models of mice to investigate natural selection. He paints 200 of the clay models white and 200 brown. He places the model mice in woodland in the morning. He checks the models in the evening for damage by predators. The graph shows his results. So we can see that the damaged mice are far more prevalent with the white mice compared with the brown mice. Calculate the difference between the number of damaged white mice and the number of damaged brown model mice. And the crucial thing here is number, whereas the actual graph's been given as a percentage. So we need to work out how many of those white mice were actually damaged. So if you read along the graph, you can see that 75% of the white mice were damaged. But how many were there in total? Well, there was 200. So we need 75% of 200, which is simple maths, and gives us a number of 150 mice. Let's do the same for the brown mice now. Reading off the graph, you can see that it's 28% this time. Again, there were 200 brown mice. So that is 56 mice. So if you take 56 away from 150, you get 94 as your answer. Suggest why the results would differ if the student used live white mice and live brown mice rather than clay models. 2 marks, you want 2 separate points. Well, the obvious thing to write here is that the live mice would run away, could be eaten, and could even go and hide. 4. The world supply of wild fish is decreasing. Fish farming provides an alternative method of supplying fish. The graph shows the change in total supply of fish and the supply of wild fish between 1950 and 2017. The graph also shows the predicted total supply of fish and supply of wild fish from 2017 to 2050. The total supply of fish is the sum of the supply of wild fish and the supply of farmed fish. Describe the change in the supply of wild fish and the supply of farmed fish between 1950 and 2017. It's a describe question, which means you need to say what you see on the graph and don't provide any explanation, because that wouldn't be answering the question. So as you can see from the graph, we've got a positive gradient. So you can say that there is an increase in farmed fish, and that with the wild fish that they increase in number and then decrease. 2. Calculate the predicted rate of increase in the total supply of fish in tonnes per year between 2017 and 2050. Give your answer in tonnes per year and make sure you show your working. So let's mark on our graph how we're going to do this. So we need to look up 2017. So bring that up. It's the total fish that we're after. So I'm bringing that line all the way up to here. This is 2015. And then we need to use our scale on the y-axis in order to do this. And look at the difference between 220 and 160, and that's obviously 60 tonnes. And we're looking for our answer in tonnes per year. So we know that 60 tonnes was made in 33 years. So how many tonnes is that per year? So we do 60 divided by 33 to get 1.818. To increase production, fish farmers maintain water quality and food quality on a fish farm. Explain 3 other ways that farmers could increase production on a fish farm. So we need to discuss things which haven't already been mentioned. So the first thing to mention is that the farmers feed antibiotics to the fish. And why do they do that? That's in order to kill pathogens which might otherwise cause disease within the fish. The next point you want to make is that fish farming obviously often uses nets in order to keep those fish in place, and the other real reason is to stop predation, so to stop predator birds coming in, swooping in and eating the fish. So nets protect fish and prevent predation. And lastly, when we're looking at fish farming, they often use selective breeding programs, and that's to ensure that the fish which are grown have a high yield, so that there are plenty of them, or that each individual fish has a high mass, because a fish farmer wants big fish. They don't want a fish that's very small that won't get that much money at market. So selective breeding programs increase the yield of fish. 5. A student investigates the effect of deforestation on the number of insects living in the soil. She uses a quadrat. That's going to be key. Remember, that's that wire rack that you put down on the ground to sample the soil from 2 areas in an oak forest. One area has trees growing, the other area has been deforested, so that means the trees have been cut down. She compares the number of insects in each area. Her table shows the results. So trees growing, we can see that there's a huge number of insects per metre squared, 1,943. Where the trees have been deforested, we can only see 40 per metre squared. So name the independent variable in this investigation. So you must learn the independent variable means the thing that you're changing. So we're going to be examining areas with trees and without trees. Part B, suggest an explanation for these results. So why do the insects like the trees? Well, that's because the trees provide an excellent habitat for insects, such as trees provide insects with food and also shelter, and help them stop being spotted by predators. The student's results are valid and reliable. Suggest how the student made sure that her results were valid and reliable. So reliable is all to do with being able to repeat and get the same results every single time. So we're repeating, which means that the student must have used several quadrats. And remember, when you're placing quadrats, because this is the bit where it's on about how valid it is, it needs to be a very well set up experiment. When you place quadrats, they must be placed randomly. So student used several quadrats and placed them randomly. And then in order to make sure it is a fair test, you want to make sure you're sampling at the same time of the year, because it wouldn't be fair to do one sample in the winter and one in the summer. D. The student claims her results show that deforestation should be prevented. What are the advantages and disadvantages of deforestation? 6 marks, so try and split your answer nice and evenly. So try and think, why would we want to deforest? So let's start there. Advantages of deforestation include the fact that there's more space to grow crops. And really, the wood can be used for many different things. So the wood from the trees is used as a building material, and fuel, because you can burn it, and subsequently that will provide jobs. Disadvantages now. So many different things you can say. So when that wood is burnt, remember it releases carbon dioxide, which is a greenhouse gas. Because you've cut down trees, it means that transpiration is reduced, and that affects the water cycle. And the third point is, as we've already seen from the insects on the other side of the page, woodland provides huge habitats for insects. So if you cut the trees down, you have a loss of habitat. So try and notice that I have made 6 very separate points here. Don't just keep repeating the same point. They won't give you more than 1 mark for that. 6. One of the largest land mammals ever known is an extinct species of rhino that lived in Mongolia. The diagram compares the size of the extinct rhino with an existing rhino. My goodness, that is so much bigger. And our magnification for this is times 0.01. Using the scale on the diagram, calculate the shoulder height of the extinct large rhino. Give your answer in meters, and you want to use this formula triangle, IAM, where I stands for image size, A stands for actual size, M stands for magnification. So we're looking for the actual size of the rhino, so we need to do the image size over magnification. So we know that's 0.01. So you need to get your ruler and measure the length of this, and if you've printed it out properly, you should measure that, and it should be 5.6 centimeters. Because they want the answer in meters, we need to convert that into meters by dividing by 100, so it becomes 0.056 meters, and then just pop that into your equation down here. And once you've done 0.056 divided by 0.01, you get an answer which is 5.6 metres. B. Some people think that the large rhino became extinct because of global warming. Suggest how global warming could have made it difficult for the large rhino to maintain a constant body temperature. Now, if you have global warming, it means that that rhino is going to suffer, because as you can see from its size, it has a very small surface area to volume ratio, and that means that it won't be able to cool itself down as readily. So let's write that here. The large rhino has a small surface area to volume ratio, which means that it is less efficient at cooling itself. Suggest 2 factors other than global warming that may have caused the large rhino to become extinct. This is a nice easy question. So what on earth could have killed that rhino? Well, it could have been pathogens, so spread of disease. It could have been loss of habitat, which you might have had a clue from the previous question. So something like deforestation would lead to habitat loss. It could even been a natural disaster such as flood or landslides. So let's make a note of those points now. So disease and also habitat loss are good options. You could have also mentioned that they could have been predated upon by humans. Seven, all living cells contain enzymes. Describe the role of enzymes in cells. So they're really asking you for the definition of an enzyme, which is that it is a biological catalyst. And you do need to learn these off by heart, which speeds up the rate of reaction without being used up. The table lists some enzymes, where they're produced and their function. Complete the table by giving the missing information. So amylase, where is it produced? The salivary gland. What is its function? To digest starch. Protease now, where is it produced? Many places, but the best place to state is the stomach. Remember that it digests proteins, as its name suggests. We're now after an enzyme found in a bacterial cell, and its role is cutting DNA at certain points. So this is the genetic engineering part of the specification. So we're looking for the restriction enzyme here. And linked with this is the enzyme given below, ligase. It's found within the cell nucleus. Well, what's its job? Remember, it was to stick the sticky ends together of that DNA. So it's going to be joining DNA. C. Explain how changes in temperature can affect the activity of an enzyme, and note that it's changes, so that could be increased temperature or decreased temperature. It's worth 4 marks, so we're going to have to write about both. So you want to say low temperatures slow enzyme activity, and that's collision theory, so it's a chemistry answer, because the enzyme and substrate particles have low kinetic energy. And therefore collide infrequently. Skipping on to when you have very high temperatures now, we also see a reduction in enzyme activity, and that's because the enzyme has been denatured, which means that the substrate molecule no longer fits inside the enzyme's active site. 8. Plant roots grow when cells in the root tip divide by mitosis. Cells in the process of dividing by mitosis can be identified because their chromosomes become visible when viewed with a microscope. The photograph shows the cells of a squash root tip seen using a microscope. How many cells in this diagram are dividing by mitosis? And you're looking for the ones where you can see the chromosomes looking like they're migrating to opposite ends of the cell. So that's 1, 2, 3, 4, 5, 6, 7. Scientists measure growth in root tips by calculating the mitotic index. The equation shows how to calculate the mitotic index. Mitotic index is given by number of cells showing mitosis divided by the total number of cells times by 100. Root tips that are growing rapidly have a high mitotic index. Calculate the mitotic index for a root tip with 9 cells showing mitosis and 110 cells not showing mitosis. Be very careful, this looks easy and it is easy, but make sure you're reading the question properly. So mitotic index is the number of cells showing mitosis, which we know is 9, over the total number of cells. So that's 9 plus 110, which is 119, times by 100. And when you put that into your calculator, you'll get a value which is 7.56. Explain why it is difficult to obtain the data to calculate the mitotic index. Well, look, we've just had to count those cells and they look pretty difficult to count. So that will be our first point. It's hard to count the cells, especially because the chromosomes are so small. You could have also said something about the microscope having a low resolving power. C. A student uses this apparatus to investigate the effect of a plant hormone on the mitotic index of onion roots. The student puts onions in different concentrations of plant hormone solution. She then squashes samples of the root tips and calculates the average mean mitotic index for each concentration. The table shows her results. Describe the results of this investigation. So again, have a look in that table, say what you see, and we can clearly see that when there's 0 concentration of plant hormone, you still get some mitosis. There is a mitotic index. So let's make that point first of all. And where does most mitosis occur? Well, it's when the concentration of plant hormone is 0.005. Name 3 abiotic, non-living factors that the student should control in her investigation. So what could make this an unfair test? Well, you need to learn these following abiotic factors, which is temperature. You must keep that the same. You could normally say the pH of the soil, but we can see from our experiment there's no soil, so don't really put pH. That doesn't make a lot of sense. You could talk about the light levels. That should remain the same, as well as the oxygen levels. That should also remain the same. You could have also said the amount of mineral ions, the volume of solution, and the time. NASA, the National Aeronautics and Space Administration, I did not know that's what it stood for, is investigating how to grow plants on space stations. As part of their investigation, NASA scientists looked at the effect of using different types of lamp on the rate of photosynthesis in spinach plants. Explain why the scientists expected a link between the rate of photosynthesis and the biomass produced. While photosynthesis, we know from the equation carbon dioxide plus water produces glucose and oxygen. Glucose has a mass, and therefore biomass will increase when photosynthesis increases. So I'm going to write photosynthesis produces glucose. And then don't forget that glucose is used in the plant's respiration to release energy, and that energy is then used in the plant's growth. So let's say glucose is used in respiration to release energy for plant growth. B. The scientists measured the rate of photosynthesis and the biomass produced for different types of lamp. The table shows their results. Plot a bar graph to show the biomass produced for each type of lamp. So don't forget what this graph should look like. It should be a bar chart, not a line graph. So let's make sure we have the correct labels. So we're going to have type of lamp along the bottom. And then we're looking at biomass. So we're going to have biomass along the y-axis. And don't forget your units, which in this case is gram per metre squared. And then pick a sensible scale. So we need to get up to 9. And I'm actually going to reduce this graph line because the scale is such that we only need to get from 6 to 9. And I want to keep it nice and big. So I'm going to put that as 6, 7, 8, 9, 10. And then type of lamp, fluorescent, sodium. I've got that from the periodic table, by the way. LED 660. I'm struggling to fit this in. My pen is too fat. LED 670. LED 680. LED 690. Try and have a nice sharp pencil so that people can actually read what you've written, unlike what I've done here. So for the fluorescent lamp, we're looking for biomass produced of just being 6. Use your ruler as well. For sodium, it's 8.8. LED 660 is 7.8. 670 is 8.2. 680 is 7.8. And 690 is 9, so that's definitely the highest. And it's worth learning how to draw graphs properly because they are worth a whole 5 marks. Suggest why the rate of photosynthesis and the biomass produced do not follow a similar pattern. And that's because not all the carbohydrate or the glucose made is used for growth or to produce biomass. So you need to try and think about other places where that glucose could be used. Well, it could be used in respiration, which I've already mentioned. And remember, the plant has to carry out active transport. So when it absorbs mineral ions into its root hair cells in its roots, it uses active transport to do that. So a lot of that energy, a lot of that glucose used won't necessarily just be used to help the plant grow. Suggest which lamp would be best for a spinach grower to use in his glass house. So let's look back at the results at the table even to see which one produced the most amount of biomass. And we know the most amount of biomass was 9 here. We can see it on the graph here, and that belonged to LED 690. So we state the lamp and we say why, because it produced the most biomass. Name 3 factors other than light that would affect the rate of photosynthesis. Remember, part of the specification is learning the factors which affect photosynthesis. So that is light intensity, its carbon dioxide level, its temperature. And the other secondary ones you can mention are water or availability of mineral ions. Suggest why NASA is investigating how to grow plants in space stations. So why would we want to grow plants? Well, potentially to provide food for our astronauts or people that go and live there. And the other great thing about plant photosynthesis is they produce oxygen, which, as we know as humans, we need it for our aerobic respiration. So plants are great, which is why we shouldn't be cutting down trees. 10. The diagram shows a nephron from a human kidney. Name the structures labelled A, B, C and D. So I'm just going to go through this right now without even looking at the bottom. answer sheet. That's the collecting duct. This is the distal convoluted tubule. This is the loop of Henle or Henle. This is the proximal convoluted tubule. We have our Bowman's capsule, and we have the glomerulus, which is the blood vessel feeding into that nephron. So make sure we match up our labels. So A is obviously collecting duct. B is loop of Henle. C is the glomerulus. And D is the Bowman's capsule. B. The nephron produces urine that flows out of tube A to the bladder. Explain how changes in the composition of this liquid are brought about when a person is dehydrated or short of water. Yay, this is a nice question. So we're looking at how does our body preserve water, and we need to be talking lots about the hormone ADH. So first of all, our body needs to recognise the fact that there's low water levels, and it does that in the hypothalamus, which remember is part of the brain. So the hypothalamus detects low water levels, and then remember that it sends a signal to the pituitary gland, which is also in the brain, to release more of the hormone ADH. And that hormone, as all hormones do, travels in the blood. And it goes specifically to the collecting duct of the nephron and makes its walls more permeable to water. And as such, more water is reabsorbed back into the blood, meaning that the resulting urine is concentrated and low in volume. Human lungs and plant leaves both contain gas exchange surfaces. These gas exchange surfaces have a number of features in common. The diagram shows a cross-section through human lung tissue. Explain with reference to features shown in the diagram how lung tissue enables efficient gas exchange. So what are we really looking at here? Well, we have an alveoli here, and we have a blood capillary here. And remember that oxygen moves into the blood and carbon dioxide leaves the blood so it can be exhaled out of our bodies. So first of all, you must mention the various parts of the gas exchange system. So the alveoli has a large surface area for gas exchange. You can also see how close together the capillary and the alveoli are, which means that there's a short diffusion distance. Similarly, the blood capillary wall is thin. And lastly, which you really can't see from the diagram, but it is a point to learn and it is a mark on the Mutt scheme, is that it's a moist environment to help gases dissolve. B. Explain how the structure of a leaf is adapted for efficient gas exchange. So remember, we've got the structure of the leaf. We have the waxy cuticle, we have the upper epidermis, then we have the palisade mesophyll, spongy mesophyll with air spaces, that's important. And then the bottom layer contains guard cells and stomata. So it is good to be able to just whack these out so you can remember them. So looking at that leaf structure then, what is its adaptations for gas exchange? I've just said it has spongy mesophyll, and be nice and scientific here. You want to say guard cells contain stomata that allow CO2 into the leaf and oxygen to leave, and then points that you've literally just made relating to that alveoli and capillary, they're relevant here. So you want to say thin with a short diffusion distance, and moist to allow gases to dissolve again. So look, you've written the same answer twice and gotten 4 marks for it. And it's the final question. So 2 different varieties of yeast, A and B, are used to produce beers that contain different concentrations of alcohol. Design an investigation to determine which variety of yeast is best for producing low alcohol beer. Your answer should include experimental details and be written in full sentences. Right, we love these questions. Corms, it's so straightforward as long as you follow this technique. And don't forget that my revision guide again has all these hints and tips to help you score full marks on these questions. So first of all, we want to state our independent variable, and that's the thing we're changing. So we're changing whether we have yeast A or yeast B. Our dependent variable is what we're measuring. So look at what we're actually studying. We're looking at alcoholic beer, so we could measure the amount of alcohol in that beer. And don't forget at this point to mention a time frame. So let's say over 7 days. So after 7 days. So important that you mention a time frame. Then we need our control variables, so everything we need to keep the same. So we will need to give our yeast glucose that it can feed upon, and that will actually, so it can carry out its anaerobic respiration and produce alcohol. So we need to control the amount of glucose, the temperature, and the pH. Anything sensible here. And then lastly, for reliability purposes, we need to repeat and calculate an average. And I promise this is applicable for every single Corms question. Right, hope you found that helpful, guys. I'm so happy I've got my iPad so I can finally write on the screen. Don't forget to like and let me know if you found it helpful, because these videos take ages to make, so I need an incentive to keep making them. And I'll be back soon. Don't forget to subscribe and come follow me on Insta, because I'm adding lots of interesting videos daily.